Live Well, Developments within Gene Therapy, presented by Dr Jasleen Jolly, Anglia Ruskin University, Cambridge, on the 13th of January 2022. About Live Well. Live Well is a series of events aimed at helping people make the most of life with sight loss. Live Well is a collaborative project between 7 local independent sight societies. Sight Advice South Lakes, Cumbria, My Sight Notts, Nottingham, Sight Airedale, the Airedale area of North and West Yorkshire, Support for Sight in West and South Essex, Sight Concern Worcestershire, Sutton Vision in the London Borough of Sutton, and Outlookers in Huddersfield, West Yorkshire.
So good morning, I am Jasleen Jolly. I've recently moved to Cambridge and joined as an Associate Professor of Vision and Eye Research at Anglia Ruskin. I'm going to share my screen because I've got a few slides to show you and I will try and describe them as I go along. These slides are showing Oxford because this work was done in Oxford. I used to work in Oxford University on the gene therapy programs, and I'm trying to bring gene therapy into Cambridge. So that's one of the reasons I was hired here. And I've joined a partnership with some of the local people here. So to bring some of these treatments that I've been working on, and I've worked on not only gene therapy, but various other treatments, but I'm going to talk about gene therapy today, and I can always come back and talk about some of the other treatments at another time.
Please do.
So why is this all important? It's because it's the future of medicine. So what I'm showing here is a selection of news reports as well as academic papers about how gene therapy is successful, and the FDA approved the first gene therapy as a clinical treatment, and it is now has been approved for use on the NHS, and we've seen the first treatments for Leber's congenital amaurosis, which is quite a severe form of blindness and occurs from quite an early age, and it is being treated on the NHS using gene therapy. So this is the first eye condition which is being treated with gene therapy. So it's not even science fiction anymore. It's happening right now, which is really exciting. And this opens the gateway for other therapies. There are over 50 conditions that are being trialed with gene therapy at the moment across the world, many of them in the UK. So the current hubs for gene therapy are Oxford and Moorfields. The current genetics centres, specialist centres, are Bristol, Manchester, Oxford and Moorfields, and Cambridge is now becoming one of those as well, which are on the screen. So yeah, basically gene therapy is already happening, but it's in trial for lots of different conditions now. And what I want to talk through to you today is the process of how we get there. But first of all, I thought I'd share some words from one of my previous patients from Oxford who took part on one of our gene therapy trials.
At the operation?
Well, since we've had the operation, it's definitely improved. There's a huge difference in the clarity between the two eyes. I can describe it as looking through a very dirty spectacle lens, or the colours are very much washed out in the untreated eye, but the treated eye is very, very clear.
And how has that impacted on how you've been able to function on day-to-day tasks?
It makes moving around, I suppose, sometimes easier, but it's just almost a pleasure to notice that difference now between the two and kind of what I've missed out and been missing out on for quite a long time.
And I think you mentioned about your night vision.
That's still there. So since last year, when I noticed the stars at night, I do keep checking on a regular basis that I can still see them, and definitely in places where there's less artificial light, it's quite a noticeable difference. And between the two eyes, I can see stars with my left eye, treated eye, which disappear when I cover it up and look through the right eye.
So how do we get there? Well, we have a huge amount of work to get to that stage. We need to do preclinical work. So that's testing in animals. So first we need to figure out what the gene does that isn't working. What is that protein doing? How does that protein work? How big is that protein? How is that protein constructed? What do the defects in the protein do? Like, what impact do they have? Is it a matter of how it's folding? And then when the protein's defective, is it just that the proteins, when the gene's defective, sorry, is it just that the protein isn't made, or is it made and having a toxic effect? Because all of those have implications on when you put in a replacement gene. Because if it's having a toxic effect, just putting in the replacement gene isn't going to be good enough. You would then also need to knock down the toxic protein. And then once you make your construct, so your new protein, so your new gene, which healthy copy, which makes the healthy version of the protein, you need to make sure that actually expresses it works okay, and it's produced in an amount enough to protect the cells that are left. Then you need to go into human clinical trials, and you have multiple phases of human clinical trials. So you have phase 1, which is for safety, phase 2, which is looking at how effective the treatment is, phase 3 is when you basically create the end product in a factory that would be employed to create that final product, and then you need to test that product. So it's not using the research grade anymore, but it's using what would be used in the treatment, in the therapy by the other doctors to make sure it works. And it needs to be done in lots of centers across the world to make sure it's not just down to the individual surgeons that it's working. Then you need to put in the approvals to the regulatory authorities. So this is the FDA, the Food and Drug Administration for the US, the EMA for Europe, and now since Brexit, we also need to go to the MHRA for the UK, and then NICE decides on what's being funded on the NHS. So it's a really long process. And they look not just at how effective the therapy is, but they also look at the costings of all of it and the benefits versus the risk. And before you can do the clinical trials, where a lot of my work comes in, is looking at how do you measure the effect of the therapy? Because when you do the animal work, you can cut up the eyes and see how much protein, like directly measure how much protein is being produced. Obviously, we can't do that in human eyes. So we need to indirectly measure what the vision is doing. And that's where my role comes in. So I need to come up with tests to be able to measure what the vision is doing. And because we're looking at rare diseases that often have been ignored because they had no previous treatment, a lot of our standardized tests aren't directly measuring the changes that we want to look at. So I need to come up with new ways of looking at the vision, either through imaging or different ways of measuring the vision, such as measuring night vision, et cetera, et cetera. So a lot of background work has to go into doing these clinical trials, because you can have the best treatment in the world, but if you can't prove it, it's not going to get approved as a treatment. So you need to do a lot of work to even get to the clinical trial stage, and a lot of research studies which don't always have direct benefit for the individuals taking part before to make sure that the clinical trials are going to be effective. So the next clip I'm going to show you is from a BBC News clip that came in, filmed one of our research participants on their journey, and it can explain how the gene therapy works.
Reports.
Jonathan Wyatt is going blind. He can just about see enough to work from home. But 10 years ago, he was a successful courtroom barrister. He had to abandon his job because he began making mistakes.
The worst occasion, I think, was when I was reading out a statement to the court and I made a mistake. And the judge turned to me and snapped, "Can't you read, Mr. Wyatt?" I then decided it was time to put my wig down and leave advocacy.
Now, have you got any specific questions about what we're going to do?
Jonathan is losing his sight because of a faulty gene. He's the first patient in the world to take part in an experimental operation that aims to replace the faulty gene in his eye with a working one.
What we hope is that by putting the gene back into the cells, we can stop further degeneration and obviously preserve the vision that he already has.
In Jonathan's case, because of a faulty gene, cells at the back of the eye have been dying, leaving only the ones in the middle. Doctors hope to stop further degeneration by injecting new working copies of the faulty gene to keep the remaining cells alive.
Jonathan's operation is just about to begin. There's a little bit of tension in the air because it's an incredibly precise and delicate procedure. One small slip and his retina could tear, and the attempt to save his sight will have failed.
The procedure has never been carried out before. Now, the riskiest part, as the needle is inserted by a foot-operated control. So has the operation been successful?
It went very well indeed. Absolutely no problems.
So far, so good.
So far, so good. Yeah, no problems.
Jonathan claims he can already see better.
Left.
But it's far too soon to be sure whether there's a real lasting improvement. If it does work, gene therapy could be used to treat a wide variety of sight disorders.
More left.
Palab Ghosh, BBC News,
Right.
Oxford.
So I've actually seen Jonathan for 10 years now, because I joined this project soon after he had his operation. And I can confirm the benefits he got from the surgery have lasted all this time. And the treated eye is now better than the untreated eye. So the next slide I've got on here shows a triangle, and it shows so what I call the gene therapy triangle. So the three things you need for successful gene therapy. So one part of the triangle I've labeled the viral vector. So this is how we deliver the new gene. The next aspect is the surgical delivery. And then the third side of the triangle is the therapeutic effect measurement. So I'm going to talk about each of these. So the viral vector, the surgery, I've got a, is that not playing? For those of you who can see, this is a video of the surgery. It's we've now got a robotic arm that's doing this surgery, where it's a tiny needle and then lift up the retina to inject some fluid with containing the viral vector underneath the retina, and then the retina flattens down. So there's a limit to how much of the retina we can treat, because you can't lift up too much, otherwise it won't settle back in the right place. And what we're using is an AAV virus with all the virus DNA taken out, and then the human DNA containing the gene that we want put in. So there's a limit to the size of the gene. So not all genes can be delivered using this method. So the next part is looking at how we measure. So whether this is imaging or we're measuring some kind of visual function, we need to look at the patterns of what's happening to that vision. So, and the pattern is more important than just the results. So we need to look at patterns over time, because if you don't put enough in, so I've got an image here of cells. Each of these hexagons is a cell. A black cell represents a cell that dies, and a green cell represents a cell that stays alive. So in this, where you've got not enough viral vector has been injected, 2 green cells stay alive, but then there are lots of black cells, so lots of cells dying off because you've not got enough protein to protect those cells to survive. So you might get an initial improvement in the visual function, but then slowly as the cells are dying off, you get a decline in that visual function over time. Then if you overdose, you'll get a sharp increase at the beginning because you get an improvement at the beginning as everything improves. But because you get a toxic effect from having too much of the viral vector in there, more of the cells die off. So as the cells go black because they're dying, then the vision is also going to tail off. So you get a sharp decline from that overdose and a tailing off of the vision. If you get it right, then you might get a little bit of an improvement. We're not actually looking for an improvement, we're looking for stabilization. Any improvement we get is a bonus because we're only rescuing the cells that might be on the edge, and we manage to rescue them from the point of no return, like just before they reach the point of no return. You might get a small bit of decline in the long term because there's some cells on the edge that will still go, but overall we'll get a steady state, which is less than the natural decline. So this is why we do natural history studies, so we know what the normal rate of change is in any disease. And natural history is so important to measuring therapeutic effect. So this is what we really look for when we do, when we're looking at therapeutic effect, is looking at those patterns over time, and that's absolutely critical. We do have to be a bit careful though, because visual acuity, you know, when you go and read the letters on the chart, that can be a bit misleading because that literally only represents a few cells at the very center of the back of the eye. It represents literally 0.5 degrees at the center of your eye for those letters. So if those few cells at the center of your eye happen to be the ones that die off, you can get the dip in that visual acuity measurement, but everything else stays stable. That still is a successful treatment. So it's important for us not just to look at the letters, but to look at a multitude of measurements. Otherwise, we're not going to get an accurate definition of what the treatment is doing. A lot of people get really hung up on the letters, but the letters are only a small part of the story because they represent so little of what your retina is doing. Also, the letters can present difficulty. I don't know how many of you have retinitis pigmentosa or similar conditions. So what I'm doing here is presenting a video which simulates retinitis pigmentosa. So here we've got a scene of the famous Oxford spires where, and we're showing how you lose your peripheral vision to give you tunnel vision over time with the retinitis pigmentosa. And when we're trying to measure visual acuity on a traditional chart, this can become quite difficult because when you've got the tunnel vision, it becomes quite difficult to track across the chart. So we don't always get accurate measures of the vision either, because you can lose your letters as you're trying to look across the chart. You lose your place. Does that sound familiar?
Mm-hmm.
I can see people nodding their head. So what we did is we did a study where we looked at using an electronic chart. So you wouldn't be having to read across, but we present the letters on the center of the screen, and thereby it makes it easier to get an accurate measure of your vision because you're not having to read across. We just present the letters in the same place all the time, so we can make the letters smaller much more easily for you. So it's studies like this that help support the clinical trials. So they have no direct benefit to the individual taking part, but they help the wider program. And they're so critical to the success of the bigger programs. So, and then we also use imaging, a lot of imaging. So when you go in to have your assessments in the eye hospital, you probably get subjected to a lot of flashing lights, probably a lot of, there's one that people often complain of, which is a bright blue light that people often don't like. So the bright blue light is the image on the right-hand side here, and that's so useful because it gives us an idea of where the living cells are, so we can actually measure those. So I've outlined where the living cells are in blue here, and we can actually measure those. And I know, so this is an image. I've got a healthy image on the left, a patient with choroideremia, which is the gene therapy trial, one of the gene therapy trials we're running in Oxford. And we can look at these images and measure them over time. And we've done studies where we've measured people over 10 years, and I can tell you what rate of decline we expect. And then after seeing patients for 2 or 3 years, I can advise a patient whether they are progressing slower than average, faster than average, and that just allows them then to plan for their life. And we're doing this in a number of different diseases, and the hope is that when the clinics get up and running in Cambridge, we can put our data together with the Oxford team, and I'm also working with the University of Melbourne with their genetic clinics, as well as with the Netherlands and with Bonnet in Germany. So by putting all of our data together, we get a lot more data, so we can do a lot of this faster, and then we can advise patients so that they can plan ahead, and give you, for those of you who want to know, we can give you a better idea of where your vision is heading. Also, by having this information, we've got more information to plug into the gene therapy trials. So when it comes to getting approval for gene therapy, again, it helps with that process, because when we do the trials, we try and include people with earlier disease, with later disease, because we don't want it just to be approved at one disease stage. And we can provide information. So if they want us to prioritize people with certain disease states, you know, we've got all this background information. So whatever the regulatory authorities ask us, we've got this information ready to hand, and we don't need to delay the process. Okay, so, and we do a lot of visual fields. So this is a technique called microperimetry. So it's more accurate than the techniques that you would have done in your optometrist or even in most hospitals. So with microperimetry, we're tracking your eye movements at the same time as doing the visual field test, which means that we're compensating to some extent for your eye movements while we're doing the visual field test, which makes it a lot more accurate. And the lights are being projected directly onto your retina. So we're measuring your retinal function directly rather than projecting the lights into visual space, which means that we're getting a much more direct measure of what's happening at the retina. And this allows us to make much more detailed assessments of what's happening. And this is used in a lot of the gene therapy trials now. I've done a lot of background work on microperimetry. It's been a big part of my career, and it's quite an incredible technique because it allows us to go into so much more depth. And I'm working with a colleague at UCL who's an expert in AI, and is looking at patterns in loss, in visual field loss over time using AI techniques. So we've looked at a number of different diseases. So the top one here is Stargardt's disease, where you get the central loss. So in this patient, I've got two types of visual field. On the left-hand side is microperimetry. On the right-hand side is a machine called the Octopus, which shows the wider visual field. And in the microperimetry, you can see that they've got very central fallout of their visual, visual field. Whereas in the wider visual field on the right-hand plot, it's all normal. The few dots at the top are just due to the eyelids, which is normal. And then the bottom plot is a patient with choroideremia. So there you can see all the peripheral visual field is lost, but the center is, they've got some normal field in the very center. And in the wider visual field, they've got pockets in the side, which are normal, and pockets in the center, which are normal, and then pockets all over, which are lost. That would make, so they're both, those patients are going to have different issues with their vision. And the genes that cause these two conditions are very, very different. So choroideremia is caused by a gene called REP1, and that's in gene therapy. The top one is caused by a gene called ABCA4, which is really common, but much harder to treat with gene therapy because it's a massive gene. Though there are attempts being made at trying that, but it's having to use a very different approach because it's such a large gene. And now CRISPR-Cas9, which you may have heard about on the news and was a Nobel Prize winning, is also being used in some ways, but at the moment it needs a lot more lab work before it can really come into human trials. And then we can combine the two measures to look at changes over time, and look at how well the structural changes and the functional changes correlate. And then we can do this kind of process in a whole host of measures where we can look at night vision, we can look at contrast, we can look at the speed of vision, color, as well as detail. And then finally, I've been moving more into the neuroscience of low vision because everything's interconnected. All the circuits in the eye are interconnected, and then the brain and the eye are really interconnected because you need both to see. You need the image, you need the light signals from the eye, and then you need the brain to interpret them. And if we were in person, I have some models of the eye that I'd let people feel. So if we get to meet in person, I will bring these with you to show you. But for now, I've got on screen, which is not as effective, but hopefully I can bring these to give you a tactile experience, because these are 3D models of my brain. So the vision part of the brain is at the back, but over 50% of the brain is connected to vision in some way. So let me show you. So on this one, I've painted on the frontal eye fields are painted on in light pink at the front, are connected to the back where the primary visual cortex is, and those are connected. And there are loads of other centers in the brain that are connected, but I really hope I can meet you in person and I can bring my tactile models for you to have a feel, so you can feel what the brain is really like with all the folds and all. So we can do visual field testing actually in the brain, and we've done this, and we're looking at this data at the moment in patients with Stargardt and choroideremia. And we've done this at 2 different time points, 2 years apart, to look at how well the two things correlate. And I'm also currently doing this following gene therapy as well, to see the effect of treating the retina on how the brain wiring is working. And the aim is to maybe harness, we know the brain is plastic because whenever you learn a new skill, your brain wiring is all changing and adapting. So the aim is to use that brain plasticity as a low vision treatment in future. And that's one of the big projects that I'm going to be working on over the next few years. And we're also we're currently recruiting to understand Charles Bonnet syndrome. So I heard you guys mention Charles Bonnet earlier. So I'm working with Esme's Umbrella. They've part funded one of my studies where we're trying to understand where in the brain those hallucinations come from, so that we can then decide how to treat them for those who find them unpleasant. So we need volunteers under the age of 70 to go in the MRI scanner for us. Full travel expenses to Oxford will be paid. So please do get in touch. If you're interested in volunteering for that, please contact my study team. The email address is brain@eye.ox.ac.uk. So that's brain@eye.ox.ac.uk. And then my team will send you more information and run through the screening questions with you. Yeah, that would be great. But until we understand where the hallucinations are coming from and what's actually happening, there's not much we can do about it. But also, once we understand what's happening and what's causing the hallucinations, we can't get more people to take Charles Bonnet more seriously either. So at the moment, outside of the vision community, I don't think it's really taken that seriously, which is a shame, because that means people aren't getting access to enough help. Epilepsy wasn't taken seriously until the neuroscience caught up with it. ME wasn't taken seriously until the neuroscience caught up with it. So this is what we need to do with Charles Bonnet syndrome as well. We need the neuroscience to catch up to get it taken seriously by the wider medical community. So please do volunteer. So with that, I'm going to stop talking and stop sharing and
thank you, Jasleen.
Take questions.
Yeah, can we open up for questions, if that's okay? And before we start the questions, Tim and I were having a little conversation. If you would like to come back and do a further talk anytime soon, please let us know if there is anything else you would like to do for this group.
Yeah, always happy to come back. And can I encourage people to put their cameras on, because I'd love to see you and interact with you.
Yeah, I mean, we have a few people who are joining us by phone, but anybody who is so, would anybody like to ask a question?
Hi, Jasleen.
Hello.
Hi. Are you doing anything around any studies with glaucoma?
I personally am not. My field is genetic eye disease. Okay. However, the Vision and Eye Research Institute, where I'm based in Anglia Ruskin University, is doing quite a lot on glaucoma. So Professor Rupert Bourne, one of my colleagues, is a glaucoma expert. So, and I know we have glaucoma clinics on behalf of Adam Brooks at the Institute. So yes, there is a lot going on for glaucoma.
Could you please maybe do an intro email to me and to this professor, and maybe I could invite him?
Sure, yes,
thank you.
Happy to do that.
And maybe CC Tim in as well, thank you.
Sure.
We will get somebody, Mark, okay?
Yeah, lovely. It's just, I think I read somewhere that they were doing some kind of something with mice trying to regrow the optic nerve.
Yeah, we can find out from him.
Yeah, that's happening in Cambridge University. Yes, that work is happening in Cambridge University. I don't think they've got anywhere near clinical trials from what I've read so far. Yes, yeah, I mean that's really complicated. This is the same problem with stem cell research, because I've seen some of the work around stem cells as well. With stem cells, growing the cells is actually easy. It's getting them to talk to the neural circuitry is really complicated. So the problem you've got is the circuits are created in the fetus, and they like photoreceptors, the rods and cones, that are light sensitive are part of the neural circuitry. They are formed from the same cells that create the brain, and those cells are set at birth and do not regenerate. They are the same that they're the same that you have throughout life. So when you replace them, having to regrow those neural connections and then having to get them to talk is really complicated. So when you regrow an optic nerve, it's all well to regrow the nerve, but then how do you then get it to talk and actually get the signals to be processed by the rest of the neural circuitry? That's the complicated bit.
Okay, thank you.
Are you doing anything about macular degeneration?
Oh, there is so much research going on macular degeneration, a huge amount of work all over the world. There is some gene therapy going on for macular degeneration, including in Oxford, both dry and wet forms. There are lots of other treatment options also being trialed for macular degeneration. There's all the nutritional work also being done around macular degeneration in the various injection types that are being trialed for macular degeneration. Yeah, I mean, the amount of work, I struggle to keep up with macular degeneration. I focus very much in genetic eye disease. But yeah, the amount of work being done for macular degeneration is phenomenal.
Thank you for such a fantastic talk. I come with two hats, really. Officially, I work for the Macular Society, but I have a very personal interest in RP because I carry X-linked RP and it affects the males in my house. So from the macular perspective, when you're talking about the Charles Bonnet research, are you looking just for people with inherited eye disease, or are you looking for people with macular as well?
Everybody, but because we're looking for people to go in the MRI scanner, at the moment we're only looking for people under 70.
Okay, I will put the word out.
Thank you, that would be wonderful.
Thank you very much.
How about PXE, which is a hereditary disease?
It is, yes. So I know there are teams working on gene therapy for PXE. I don't think it's in human clinical trials yet, but it's worth. Are you under any ophthalmology services at the moment?
Yes, I go for 6 months checkup in Colchester.
Okay, so once we get the service up and running in Cambridge, it might be worth getting referred into us because once I get things up and running here, I will be linking into the interventional trials. And I'll also be doing a lot of the natural history work and a lot of the background work for the interventional trials. So that way you'll be kept in the loop for anything that comes up.
Right, thank you.
So I will keep the charity informed with the progress on the clinics.
Thank you.
It's Terry. Jocelyn, thank you so much for a really informative talk. I can't say I understood all of it, but thank you. No, that's my fault. A lot of science goes straight over my head. But anyway, fascinating stuff. I have a genetic condition myself. I was diagnosed with Stargardt's disease as a child. I have very, very minimal sight now, so I've lost a lot of peripheral vision. I don't know what condition I have. How do I find out what I have and what the defective gene is?
You need to get genetic testing. So you need to go get yourself seen under one of the specialist services. Where do you live?
I live in South London.
So your closest service is going to be Moorfields. If you get referred to the genetics clinics in Moorfields, and then they can offer you genetic testing.
Okay, right.
Thank
you.
So Professor Webster will be the genetics, your genetics lead in Moorfields.
I mean, I should say it's quite interesting. I'm one of 9 children, and way back in the 90s, Moorfields took blood from all of for me, and I have another sister who has the same condition, and some of my siblings, because they thought it would be quite an interesting research project, and I never heard anything back from that. I assumed that from taking blood they'd be able to identify the the defective gene, but I didn't. I don't know if we fell off their records, but I didn't ever hear anything back about that, and I seem to have fallen off their register somehow.
Oh, that's odd. Maybe check with your GP whether they received anything from them.
Yeah. Okay, thank you.
Thank you, Terry.
Could I ask a question?
Yeah.
Yeah.
Yes, Sue, please.
I got tested through the genome program. So I thought I got dominant RP, but it turned out I've got recessive RP, and it's quite a rare gene called Sparta7.
Oh yeah, that is rare.
Yeah, so is there any hope for people that have got the rare ones, or is it the stem cell route to go down? I'm 65 now, and I'm at the stage now where the last 5 years I'm beginning to lose a lot of my central vision, so I can't see photographs, but I can see a little bit of text on my computer still.
So there are treatments that are being developed which are around, which are not related to the actual gene. So gene therapy relies on us knowing what your gene is. So there are treatments being developed that are agnostic to the gene. So it doesn't matter what gene you've got.
Right.
So maybe I can come back in a couple of months and talk about some of these other therapies which are completely unrelated to the gene, and some of the work I'm doing on the brain plasticity won't matter what your background condition is. It won't even matter if it's genetic or not. So I was involved in the retina implant trials, and there's some exciting work coming up on optogenetics. I've been involved in trials for electrical stimulation of the retina to try and preserve it. I've been involved in work on electronic devices. So there's a lot of really interesting stuff which isn't related to genes. Stem cells have a different approach, but the problem with stem cells is, as I said, is getting the neural circuitry to talk, but also you need to do stem cells with the gene therapy, because otherwise the stem cells will die off due to the same genetic defect again.
Right, there's been these things which I've seen on the Facebook page, RP potential treatments, and they're where they've been doing these scaffolds of false retinas, where they've been putting the cells actually in a scaffold of a type of fibre that can, be accepted by the eye, and that seems interesting.
Yeah, I think we'll have to wait and see, because that's been very few patients that they've done that in so far. So it's a matter of just waiting and seeing how that goes.
Right.
Yeah. So there's potential options there, but yeah, I'd be happy to come back and talk about some of these other things.
Okay. I mean, just one little thing before I go. I think David's the same as well. That these electronic eyes, if it gets to the stage where that's your only option, or you think it is, if they put one of these devices in your eye and then something else came along, would that mean that you couldn't go down the natural route? If you'd already had something implanted in your eye, does it damage your eye? Could it be taken out?
Yes.
Yes, that could be taken out.
No, it does cause damage, so it would limit your ability to take part in other things.
Right. Thank you very much.
Thank you, Sue. Would anybody like to ask?
I have a question. Yeah. What about structural eye conditions such as Coloboma, congenital conditions?
Yeah. Yeah, that's a really difficult one. I don't know of anyone working on that. So that's where the brain plasticity side can come in most useful, because that's more about how to utilize the information that you'll get, like how to harness the information that you're getting in. I don't know that we could alter anything in the eye because there's not much we could do to fix the coloboma.
No, it's just that, you know, I only have half an optic nerve, and it would have been interesting to know if there was any way in utero, whether there's some sort of thing that could be delivered in utero to help rebuild that, or
possibly in the future. At the moment, I don't know of anyone that's working on that, but maybe if the growing of the optic nerve for glaucoma becomes useful, that might be able to be applied in the future.
In general, Oxford and Moorfields are centres of research excellence, and also in the United States. What are there other countries like in Europe or in Asia that are doing a lot of research as well, and how much cooperation is there between centres?
Yeah, so there's a European consortium for inherited retinal disease. So there's a lot of cooperation across Europe on these. So yes, there is definitely, and as I said, you know, I've got links with the Netherlands, with Germany, with leading centres there that are doing some amazing work, and even within the UK. There are quite a few of us that work together. So I work very closely with Oxford, with UCL, with Moorfields, so with Manchester. So we all work together because it's only by working together can we move forward. So yeah, collaboration is the key. Without collaborating, we're never going to get anywhere.
Got RP, and it's getting quite bad. And also I'm in Chelmsford, which is not too far from Cambridge, so perhaps you could pass on my email to Jasleen in case I can be any help as a guinea pig.
Okay.
Thank you. That's very kind.
Tim from Science Advice. When all the work's been done, Jasleen, you know, genetic work, you mentioned about inserting the genes using a robotic arm.
Yes.
Yeah. Can it be done with the human hand, or is it?
Yeah, that's how it started.
Right.
The robotic arm has been brought in subsequently because it allows far more precision. So it's not with the human hand. So as the BBC video showed, it was the foot pedal was controlling it.
Yes.
But the robotic arm gives much greater precision. So that's why it was brought in later on, because it allows you to do a lot more delicate work. And you know, you're injecting 0.1 of a microliter of fluid. Nice. And this is very, very detailed surgery. You're literally lifting up the retina and then you're flattening it back down. You're lifting up the retina, injecting 0.1 microliter of fluid, and then flattening the retina back down. It is highly precise work. So to reduce the risk of any side effects, the robotic arm just allows you to have much more precision.
Anybody else?
Sue had a question.
Hello, it was only very quick. It was following on. I realised I hadn't asked. Did you say, Jasleen, that for the MRI, people that want to do that, did you say there was some help with travel costs or something? I can't remember.
Yes.
I didn't write it down.
Yes,
okay.
we pay travel costs to Oxford.
Because I know that's the question I'll be asked is, well, how do I get there?
Okay. It's a single visit. It takes half a day and we pay full travel costs.
Oh, that's lovely. Thank you.
If you don't have any further question from anybody, thank you so much for coming today.
Thank you.
And thank you, Jasleen, for your time.
Thanks very much. Thanks.
Sorry again about the...
That's okay. Thank you
Absolutely
so
fine.
much, Jasleen. Lovely to meet you. And does anybody need help logging off?
Live Well and Future Vision. We hope you enjoyed this presentation. The Live Well events take place on the second Thursday of each month at 10 AM. Future Vision sessions take place on the fourth Thursday of each month at 10 AM and are technology events aimed at people living with sight loss. To attend the next session or to suggest future topics, please contact your local Sight Society, who will be able to provide you with the Zoom link.